Chapter 4, Parasitic Protozoa, Form, Function, and Classification. Form and Function, Part 2. When we're discussing the intercellular contents of protozoa, okay, we're talking the different organelles and things. Turns out many of the organelles, many, not all, many of them we have discussed before in Intro Cell and Molecular. We've discussed it before. You probably discussed it in cell biology. So many of these things that we're going to discuss, you've heard before. Heck, the way we've learned about them is we've probably studied them in protozoa, single-cell organisms. Why would we do that? Well, because the moment I remove cells from a multicellular organism, I've disturbed them. How they react to things, how they interact with things is no longer going to be under normal conditions, no matter how much I baby them in the lab. But a single-cell organism, an amoeba, euglena, something like that, it is a single unicellular organism. It's used to being alone. So, as long as I don't kill the cell, cut open the cell, it's going to do its normal metabolic functions as if it was still in a mud puddle, a stream, a lake, an ocean, whatever. So when we look at the plasma membrane, remember the plasma membrane for all cells, all living cells on this planet, is a lipid bilayer, okay? Phospholipids. Here with protists, difference is going to be which phospholipids are present. Okay, it's going to be different phospholipids for protists versus animalia versus plantae versus fungi. We use cholesterols. What form of sterols do they use as spacers between the phospholipids? That's going to be different. But for all intents and purposes, it's still going to have transport proteins, it's still going to have receptors and all sorts of things in it, that lipid bilayer, so that the interior of the cell can interact with the exterior of the cell, get things in, get things out. Difference you're going to see here with protists is the presence of what is referred to as a pellicle. Students wrongly assume the pellicle is like a cell wall. So what gives the wood, my table here, gives it its hardness. The plant cells that make up the wood, all that cellulose and whatnot, gave it this nice, hard, rigid cell wall. That's not what the pellicle is. The pellicle is not a rigid, hard wall. So, rigid, hard cell wall. What it is a protein-based meaning that there's probably going to be some sugars and other compounds in there, semi-rigid, somewhat flexible, wall encompassing the cell. It's not going to be uniform thickness, as we're going to see in this chapter and other chapters. It's not going to be solid as this, wood here. It's going to have some bend to it, some give, so you can actually see the cells alter and change their shape somewhat as they're trying to swim through, whatever water. This pellicle, it's there as a defense mechanism. It's there for protection. Remember, anything bigger than these protists, these amoeba, anything bigger than them is now predator. Okay, there are microscopic multicellular organisms, plankton and whatnot, that live off of protists. That's their food source, their prey item. So having the pellicle there makes it just a little bit more difficult for them to feed. Pellicle is also there to help with osmotic regulation. Okay, single cell organism. It is at the mercy of the salinity, the osmotic pressures of whatever it's in, the mud puddle, the lake, the stream, the ocean, whatever. Hypertonic, hypotonic, isotonic, whatever it is, this given instant can change in the next 5 minutes, depending upon where water runoff is coming from. So the pellicle will allow the cell to swell only so far. It'll allow it to shrink only so far. Another thing we're going to see with these protists that we're not used to seeing and discussing is what we see here in the right-hand picture. You have the plasma membrane, you have a pellicle, and then in between you may have some other layers of membranes enclosing different regions that are referred to as alveolar sacs. These alveolar sacs are places for storage. Storage of what? Gases, oxygen, whatever, water, nutrients, this, that, some other thing, all dependent upon what species and what that species, that cell needs at that given time point. Then we have the mitochondria. Okay, mitochondria. We all have heard these. Powerhouse of the cell. May or may not be present in these protists. The majority of them are going to have some mitochondria there because they are eukaryotes. They need a lot of energy, especially if they have cilia, flagella, locomotive mechanisms like those. The mitochondria may be exactly like we've seen before, the little bean-shaped organelle that's, multiple of them throughout the cell, or it may be some modified version of that, as we're going to see in this chapter, may be associated with, the alveolar sacs, the pellicle, things like that, meaning that its ATP production is going to be going more towards the cilia, the locomotive mechanisms that cell has. And there are some protists that have just a complete lack of mitochondria. The normal, quote-unquote normal, metabolic reactions that occur in the mitochondria for the production of ATP are going to be associated with different vesicles or different regions inside the cell. We also have our friend the Golgi. The Golgi apparatus may or may not be present. If it is present, depending upon the protist species and whatnot, it may be referred to as a dictosome. Why the different name? Well, because it looks a bit different than what we know of as the Golgi that we would see in mammalian cells or plant cells. Slightly modified organelle with slightly modified metabolic functions. Side note here, these dictosomes do not carry out the same work that we would see associated with the normal Golgi. It's because, and we know this because what they're associated with. They're associated with what we call parabasal bodies. Okay, parabasal bodies are the protist version of the basal bodies that you would see, at the base acting as the foundation for flagella and cilia. These parabasal bodies are going to be composed of different dictosomes and what are also referred to as kinetosomes. This is the entire organelle, organelle-like complex of structures, all there at the base of the cilia and the flagella for the production of ATP, for the production of and maintenance of the cilia, the flagella, and whatnot. And how do we know this? Well, if you look at these pictures right here. To the one on the left, you see that's the artist rendition of, here's pretty much what we see when we compare a bunch of different photographs, transmitting electron micrographs, which are on the far left there. You'll notice that there's multiple structures there. Multiple structures that come together. Some of them are going to be the dictosome, which is that modified Golgi-like apparatus. But it's associated with all these other structures that are responsible for the locomotion of the cell, the maintenance of the locomotion apparatus and all of that. You can also see different small vesicles or small vacuoles that have different roles for the cell. They're there for storage, but a storage of what? Peroxisome, hydrogenosomes. These microbodies, things are produced metabolically and then stored in these lipid bubbles inside the cell as a storage mechanism, because whatever's in them you don't want floating around because it could damage the cell itself. But these could be for defense mechanisms. They could be for aiding in breaking down whatever food particle was phagocytized by that cell. And they're going to have certain vesicles that are there for the extrusion, exocytosis of. They call these extrusones, okay? Depending upon what's in the extrusome will tell you what its generalized function is. You have toxosomes, kinetosomes, haptosomes, kinetocysts, haptocysts, trichocysts. Their jobs are for being extruded out under certain conditions to fight off predator, to immobilize prey, because remember, for many of these single-cell organisms, anything smaller than them is prey, so they would go and they would eat other protists. So you have to be able to immobilize them long enough to get a hold and then phagocytize, pull them in. Then you also have mucocysts, like their name sounds. It's for making of mucus, mucor, a nice little coating, slime coating that makes it hard for other things to grab a hold. Also protects them against water loss as well as chemicals getting in and damaging the cell.